Hello, I am Anuradha Malik. In the previous episode of class 11 biology, we studied about mitosis. Today we shall study about meiosis, which is another special kind of cell division. Meiosis is also said to be reductional division, which occurs for gametogenesis and results in the production of 4 daughter cells. The meiotic division involves 2 sequential cycles of nuclear and cell division called meiosis I and meiosis II, but there is only one cycle of DNA replication. It involves certain other features such as pairing of homologous chromosomes and recombination between them. The events of meiotic cell division can be grouped under the following phases: prophase 1, metaphase 1, anaphase 1, telophase 1. Then meiosis II, prophase 2, metaphase 2, anaphase 2 and telophase 2. Meiosis I starts with prophase 1, which is longer and more complex than the prophase of mitosis. On the basis of the chromosomal DNA, this phase is further subdivided into 5 categories, namely leptotene, zygotene, pachytene, diplotene and diakinesis. During leptotene, chromosomes gradually become visible under the light microscope. Compaction of chromosomes continues throughout this stage. This is followed by zygotene, during which chromosomes start pairing together, a process called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs reveal that synapsis is accompanied by formation of a complex structure called synaptonemal complex. This complex, formed by a pair of synapsed homologous chromosomes, is called a bivalent or a tetrad. The first 2 stages of prophase I are relatively short lived. During the third phase, pachytene, the tetrads are more clearly visible under the light microscope. They remain in this stage for longer, maybe for days. Appearance of recombination nodules is characteristic of this stage. These recombination nodules are the sites where exchange of segments, that is, crossing over, occurs between the non-sister chromatids of homologous chromosomes. This crossing over is the exchange of genetic material between 2 homologous chromosomes. This is an enzyme mediated process and the enzyme involved is called recombinase. By the end of pachytene, the recombinant chromosomes remains linked at the sites of crossing over. Dissolution of the synaptonemal complex marks the beginning of the next stage called diplotene. During diplotene, the homologous chromosomes of the bivalent separate from each other except at the point of crossover. These points of attachment forming X-shaped structures are called chiasmata. In the cells of some vertebrates, diplotene lasts for months or even up to years. During this phase, the chromosomes decondense and engage in RNA synthesis. In some species, the chromosomes expand enormously, producing structures called lamp brush chromosomes, which resemble the filament of bulbs. Thus, they are called as lamp brush chromosomes. Diakinesis is the final stage of prophase I. It is marked by terminalization of chiasma. Chromosomes are fully condensed and the meiotic spindle is assembled to prepare for the separation of the homologous chromosomes. By the end of this phase, the nucleolus and the nuclear envelope also break down. Diakinesis in fact represents transition to metaphase. Look at the pictorial depiction of prophase I. Notice that these are 2 dyads, meaning there are 4 chromosomes and 2 pairs of chromosomes. In metaphase I, the bivalent chromosomes align on the equatorial plate. the microtubules from the opposite poles of the spindle attached to the pair of homologous chromosomes. The centromere of each chromosome in a pair is directed towards the opposite poles. Look at this picture of the metaphase. In this diagram, two homologous pairs of chromosomes at the metaphase plate are depicted. In each pair, one chromosome is lying towards one pole and the other towards the other pole attached to the spindle fiber. Anaphase 1. The two members of each bivalent seem to repel each other, separate and move towards the opposite poles. The sister chromatids remain associated at their centromeres. Thus, each pole receives only half of the number of chromosomes or the haploid set of chromosomes. The movement of chromosomes is brought about by the shortening of spindle fibers, as depicted in this diagram. Telophase 1 involves the formation of nuclear membrane by the endoplasmic reticulum around the groups of daughter chromosomes at the opposite poles and the appearance of the nucleolus. The chromosomes in many cases do disperse, but they do not reach the extreme extended dispersed state as in the interphase nucleus. Cytokinesis follows this, resulting in a dyad of cells. The stage between the two meiotic divisions is called interkinesis and is generally short-lived. After telophase 1, this is how the cells would appear. Meiosis 1 is followed by the second meiosis division or meiosis 2. The second meiotic division is essentially similar to mitosis if mitosis takes place in a haploid cell. It divides each haploid nucleus into two haploid nuclei. Meiosis 2 can also be described in 4 phases, namely prophase 2, metaphase 2, anaphase 2 and telophase 2. Prophase 2 is initiated immediately after cytokinesis, usually before chromosomes have fully elongated. By the end of prophase 2, the nuclear membrane disappears and the chromosomes again become compact. Spindle formation is initiated and takes place during this phase. Prophase 2 can be diagrammatically depicted as shown here. Metaphase 2 following prophase 2 is the stage when the chromosomes align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of the sister chromatids. Metaphase 2 in the two cells can be diagrammatically depicted as shown in this picture. Anaphase 2 begins with the simultaneous splitting of the centromere of each chromosome, allowing the sister chromatids to move towards the opposite poles of the cells. Anaphase 2 can be diagrammatically depicted as seen in this picture. In telophase 2, the two groups of chromosomes that have reached the opposite poles once again get enclosed by a nuclear envelope, which is formed by the endoplasmic reticulum, and the nucleolus also reappears. The diagrammatic depiction of telophase 2 would look something like this. Cytokinesis follows this, resulting in the formation of a tetrad of cells, that is 4 haploid daughter cells. Before we conclude this lesson, the significance of meiosis. Meiosis maintains a definite and constant number of chromosomes in the sexually reproducing species from one generation to the next. It also increases the genetic variability in the population of organisms from one generation to the next. As you know, variations are very important for the process of evolution. Have a look at this picture of meiosis taking place in animal as well as plant cells. Students, by now you should be able to point out the differences between mitosis and meiosis. Let's write them down. In mitosis, one division completes the process, whereas in meiosis, 2 divisions are required to complete the process. In mitosis, chromosomes do not synapse. In meiosis, Homologous chromosomes synapse in prophase. In mitosis, homologous chromosomes do not cross over, whereas in meiosis, homologous chromosomes cross over. Centromeres divide during anaphase in mitosis, whereas in meiosis, centromeres divide in anaphase 2, but not in anaphase 1. Daughter cells in mitosis have the same number of chromosomes as the parent cell. A diploid cell gives rise to a diploid cell, whereas in meiosis, daughter cells have half the number of chromosomes as the parent cell. A diploid cell gives rise to 4 haploid cells. In mitosis, daughter cells have the same genetic information as the parent, whereas in meiosis, daughter cells are genetically different from the parent cell. Meiosis results in growth and replacement of worn out cells and repair. Meiosis results in the formation of sex cells or gametes. After today's lesson, you should be able to answer these questions. First, bring out the differences between mitotic metaphase and metaphase 1 of meiosis. You could support your answer with diagrams in a cell in which the diploid number is 4. Also, differentiate between the mitotic anaphase and anaphase 1 of meiosis. Practice this in a cell where 2n number is equal to 6. So, today's lesson we studied about meiosis, and in the next episode we shall learn about biomolecules. Thank you.